first, just to review what I talked about earlier in the week, because it's really important, especially when I go on to talk about today's topic, which is basically cancer and what happens when homeotic genes don't function properly. So just to review, we have homeotic genes or Hox genes. These are like the blueprint. So like if you're a future architect or whatever, you always have a blueprint for the building. So like, you know, where the bathrooms are, where the living room is, the kitchen, whatever. And so the Hox genes are the overall blueprint, like for the body. And you can see how homeotic genes are reflected in all animal body structures, that we all have the same kind of homeotic genes in order that we match, even though we're much different from a fly. But one thing I wanted to clarify, I think I mentioned it earlier, but I want to make sure I clarify. This is for animals that are called bilaterally symmetrical. So bilateral symmetry, meaning if you draw a line down the middle, then each of the sides, which is where the lateral comes in, the left side and the right side are mirror images of each other. You would with a housefly. I can't because it's positioned wrong, but if you looked at the housefly from the top down or from the bottom up, not from the side, and you drew the line, it would be mirror images of each other. That's called bilateral symmetry. So why bilateral symmetry is really important is because when the animal kingdom was evolving, when you had all these animals evolving, bilateral symmetry allowed for something called cephalization to occur. Now, cephal means brain. So cephalization is basically the process of brain development. So if you think about jellyfish, for example, jellyfish looks like that, not bilaterally symmetrical. This has something called radial symmetry. And I know I'm already off topic, but that's okay. Radial symmetry. Radial symmetry doesn't allow for like cephalization, because where's the brain going to go? The reason why bilateral symmetry is such a bigger advantage is because you have a definitive or a very distinct head region. This is up towards the top, and a distinct tail or posterior section, which is the bottom. This allows for all of the brain type stuff to develop in the what's called the anterior or the head region, because this is where the eyes are. This is generally where like the mouth, the nose, the ears, all of the sense organs are focused and located in this anterior or the head region. And that is because of bilateral symmetry. Think about most animals that, yeah, great artistry, I know. Most animals that walk on all fours, their head region is the one that looks forward. And so that's how they look for food. So that's where all of the sense organs are going to be concentrated. That's where you're going to have a concentration of nerve endings, because that's where all the information processing occurs, is in that area. Okay, and then yeah, we looked at Drosophila. Now the homeobox. So if the homeotic genes or the Hox genes, if they are the overall blueprints, then each homeobox basically represents a body part. So in your overall blueprint, the homeotic genes is the overall blueprint, but your homeobox is where specific rooms go. And it's important because if you're building a house, or if you're designing a house or an office building, whatever, you wouldn't necessarily think, I'm going to put the bathroom as the first room when you enter the house, right? That doesn't make any sense. Just like if our brains are the sensory information processing center, you wouldn't put that, for example, like in the tail region, because that's at the end of the animal. That's not like looking forward. So the eyes, the mouth, in the case of insects, you have antennae, which stick out, which can sense chemicals in the environment. So that kind of is their version of smell. All of that is concentrated where the brain is, because that's the direction that the animal travels in. That's the first thing, that's the first body part that goes forward. So each homeobox is kind of responsible for each body part. Now, I showed you what happens if there's a mutation. Again, remembering that like 80% of our DNA is just not transcribed or translated at all. Any kind of mutation, specifically if it happens in a homeobox, can have devastating consequences. So a lot of times you see these birth defects in human beings and in animals, like if somebody is born without an arm or without legs or things like that, those are results of mutations in the homeobox. Because again, even though 80% of our DNA is not translated, the homeobox areas are specific areas that absolutely code for where body parts go. So it's really, really important. If you have a mutation, for example, where let's say the liver is going to form where the heart is, then that is going to result in a miscarriage during the pregnancy. Or I guess the official name is gestation. So for humans, we call that pregnancy. But for animals, we call it gestation. I think that's another way that we use language to kind of put ourselves above the animal kingdom, even though we are not. We are animals, just like the fruit fly here. So yeah, depending on how severe the mutation is, the gestation or the pregnancy might not come to term and might not form fully if it results in something that's lethal. Lethal meaning deadly, right? We cannot function if, for example, let's say our brain is in our intestinal area. That's just, that doesn't form a human being. So that wouldn't form a human. These are examples of mutation, again, in the HOXD13 gene. So one of the important aspects of the Human Genome Project was for us to be able to identify these exact type of areas, so that if we can use things like CRISPR, I might try to find an easy article for you guys to read about that. It's absolutely fascinating. It's basically, I don't remember, what EP was it? The first time I heard of CRISPR was when I first started asking students to do science news projects like you guys did a couple weeks ago. And there was one student, was that EP 12? Somewhere around like 12 or 13 maybe. Calhoun, she did an article about CRISPR, which I had never heard of. It was pretty new at the time. So this is maybe like 5 years ago. And it's basically DNA editing. That's right. We can actually edit DNA now. So if we know the mutation in, if we know where the HOXD13 gene is, like which chromosome it's on, and we know the sequence for normal development of fingers and toes, we can actually identify where the mutation is occurring and what, and possibly how to edit the DNA sequence so that it is quote unquote normal, right? So that, I think, is the future of gene technology, is being able to edit genes. Now, a lot of people think that that's really dangerous because you might get into what's called designer babies. Like if we're able to edit genes and we know where the sequences are for all of our genes, that you can have, let's say for Thai people, you like to have very light colored skin, blonde hair, blue eyes, all of that you could actually design your own baby. And when you get into religion, and a lot of people are like, well, that's not what God intended. So I leave that up for like governments to decide, or ethics professors or whatever. Personally, I think it's very useful for curing or treating diseases. So I don't see anything wrong with that, but that's just my opinion. All right, now moving into the where gene expression can go wrong. The first thing I'm going to talk about are something called oncogenes. Okay, an oncogene is a gene that causes cancer. In fact, the prefix onco means cancer. If you go to a doctor that specializes in cancer, that is an oncologist, right? So ist, like biologist, someone who studies biology. So an oncologist, somebody who practices or studies cancer. Now, oh, that got cut off. I'll just write in the rest oncogenes. Now, we're not really born with oncogenes. They actually start as normal genes that we call proto-oncogenes. I think the prefix proto means before, but I have to double check that. I'm not sure. Where's my phone? You can just Google that real quick. Proto, because it reminds me of like prototype is like the before, like the first version of something. Proto meaning. Original or primitive. There we go. So not before, it's original. Okay. So proto-oncogenes, which are the normal versions, they are responsible for controlling your cell's growth and differentiation. When they function normally, they code for proteins that regulate the rate of the cell cycle. Remember, the cell cycle is interphase, and you've got mitosis there. So I didn't do that very well, but, and then interphase, you have G1, S, and G2, and then you go into mitosis and cytokinesis. So basically the rate of that cycle, how often cells divide. So ultimately they control the cell's growth and division. So a mutation in a proto-oncogene can end up resulting in proteins that trigger cell division to occur too often. So instead of possibly like at the end of the life of a cell, excuse me, it's supposed to exit and go into G0. If you have a mutation and the proteins just keep triggering the cells to divide, they don't ever go to G0 and they keep dividing. That results in something called a tumor. Now, a tumor doesn't necessarily mean cancer, so I'll get to that distinction in a second. I don't know, so I'll cover it up. So a tumor is an abnormal increase of cells that results from uncontrolled cell division. So it's basically a mass of cells that should not be there. Sometimes they're very small, sometimes they're in areas of the body that don't cause too many problems. Maybe they're growing, maybe they're not growing. And so there's like a lot of gray area. There's a lot of nuance in whether or not the tumor is like really, really bad, or it's not that bad at all. So that's why I was saying before, when it comes to cancer, there's so many different factors involved that you can't just say like, oh, well, if I have a mutation in my proto-oncogenes, then oop, that's it, I get cancer. No, because your body has ways of keeping in check all the processes that are occurring. So this is an example of what's called a benign tumor. You can see that the edges are quite smooth and the whole tumor itself looks smooth. They give you the size here. So this is 2 centimeters. So this looks like it's maybe about 6 centimeters altogether. So in a benign tumor, all of the cells stay within this mass. Compared to a malignant tumor results in cancer, because in a malignant tumor, the cells that are in here that are dividing uncontrollably, they don't stay within this tumor. They're going to leave the tumor. They might break off or get shed, and they're going to travel through the bloodstream. They're going to go to other parts of the body, and they're going to cause tumors to grow in more parts of the body. And that is when cancer is officially diagnosed. So it's possible to have a tumor, but not have cancer if it's benign. Now, whether it's a malignant or a benign tumor, surgery may be an option. It completely depends on where in the body the tumor is growing. The tricky areas tend to be the brain. Depending on where in the brain it is, how deep in the brain it is, surgery might still be an option. They might also use chemotherapy and or radiation. So the difference between those two, chemotherapy is injecting, I'll just say poison for lack of a better word, is injecting poison into the body through the bloodstream. So you're hooked up to an IV and they give you the bag of these chemicals that destroy the tissue. So it does usually shrink the tumor, makes it get smaller, but it also destroys normal tissue. So that's why in chemotherapy, oftentimes you see patients are very, very sick after chemotherapy treatment. A lot of times they vomit. They start to lose their hair after a certain amount of time. I don't know if you remember, and teacher Vanessa went through this a few years ago. Another option is something called radiation. That is a more targeted type of treatment where it's kind of like, it looks kind of like an X-ray, but they use radiation to target the tumor, which also results in shrinkage as well. Now, this should say tumor suppressor genes. So this is one of the ways that your body keeps cancer in check. It's quite possible that at any point in your life, you do have cells that are dividing uncontrollably when they shouldn't, but your body has a way to keep those numbers in check. So tumor suppressor genes act as brakes to suppress tumor formation. That's exactly why they're called tumor suppressor genes. To suppress means to prevent something from happening. So these code for proteins that prevent uncontrolled division. Sorry about the text is all screwed up. I think when I wiped my computer, I forgot to put, like, I used to download all kinds of like interesting looking fonts, and I think I forgot to put all the fonts back on. So that's why it's all weird format. Anyway, so even though you have proteins that regulate the cell cycle, even if those mutate and they trigger the cell cycle too often, you have other genes that code for proteins that make sure that the regulatory proteins are working properly.
Cancer is a disease in which cells divide uncontrollably. Over the past 30 years, detailed studies have shown that genes, such as tumor suppressor genes, are often destroyed by DNA mutations, allowing cancer cells to grow. In recent studies of certain breast cancers, it has been observed that parts of the DNA that control tumor suppressor genes are frequent targets of cancer mutations. First, let's explore a normal cell, which is not dividing. Inside the nucleus of the cell, transcription factors are constantly scanning the DNA for their binding sites. The transcription factor moves along the double helix until it encounters a place where the nucleotide pattern is in the exact sequence for it to stick. Once the exact sequence is found, the transcription factor changes shape and binds tightly to the DNA. This binding allows the important tumor suppressor gene to be expressed. Many proteins then stick to the transcription factor and form a protein machine to make RNA for the tumor suppressor gene. This production prevents the cell from dividing. Now let's explore a cancer cell. Like all cancer cells, this cell divides when it is not supposed to. Inside the nucleus, the transcription factor searches the DNA for its binding site. Different types of cancer have unique mutations. In this specific type of breast cancer, the binding site has been mutated. The altered nucleotide pattern is not recognized by the transcription factor. It passes by without sticking. Without the transcription factor binding, the gene stays inactive and the critical tumor suppressor gene is not expressed. The cancer cells continue to divide without control. Over time, more mutations accumulate, causing the cancer to grow faster and faster.
So in this example, as she said,
Transcription.
the mutation here is in the binding site for the transcription factor. So this is a transcription factor for the tumor suppressor gene. If the tumor suppressor gene is never translated, you don't have that secondary check to make sure that your regulatory proteins are working properly. That being said, again, it's not like one mutation results in cancer. It's like it's a collection of circumstances. So let's say there's a mutation here, the same mutation in the binding site for the tumor suppressor transcription factors. Okay. So the tumor suppressor genes aren't expressed. Okay. But if you don't have cancer cells being formed, if you don't have proto-oncogenes that have turned into oncogenes, so you don't have proteins that are triggering cell cycle too often, then it doesn't matter that your tumor suppressor gene isn't being expressed in this particular cell. Okay, so it's a combination of a lot of circumstances. So in that example, probably explain what's happening because you don't know what granulocytes are. We do also have, as part of our immune system, we have white blood cells that are called natural killer T cells, which I'll talk about next year when we do human anatomy. But natural killer cells are basically white blood cells that just kind of patrol around our body and kill any cells that are not, let's say, behaving properly. So if there are cells in your body that are abnormal, that are functioning abnormally, then you can see in this video, you've got these smaller cells that kind of go up to and attack a larger cell. They are natural killer cells that are destroying a cancer cell. So it's yet another way that we can try to, or that our body kind of protects itself. So this is the cancer cell right here. And these little tiny things, those are the natural killer cells. You can see they surround it and then they just killed it. So how they kill it is they basically stab the membrane. Here's another one down here. And that's popped basically, I guess. I don't really know how else to describe it. So every time there's a little arrow, it's a cancer cell. And done. Right. So what the natural killer cells do is they will puncture the cell membrane, and just exactly like a water balloon, the cell explodes. So if someone does have cancer, then the tumor suppressor genes are going to be damaged. So the decrease in tumor suppressing proteins can increase the rate of cell division. Cells have 3 types of tumor suppressor genes, and all 3 have to be damaged in order for cancer to occur. So again, so many different factors and circumstances. Maybe you have a mutation in one of the 3 that's not going to result in cancer. Maybe you have a mutation in all 3 that still doesn't necessarily mean that you're going to develop cancer. I think that's supposed to be end the cell cycle. So normal cells, the cell division is controlled by several factors. 1, the cell must be receiving enough nutrition. If it is a malnourished cell, it's not going to pass. I don't know if you remember when we did the cell cycle back in, was it chapter 8, I think? There are 3 checkpoints that the cell has to pass in order for it to continue in the cell cycle. If it doesn't pass those points, then it will stay at that point in the cell cycle until it can pass. So it has to be enough nutrition. It has to be attached to something, which is one of the biggest problems with cancer cells, is that cancer cells can divide even when they're not attached to anything else. This is when the spread of cancer cells occur, and this is when cancer is officially diagnosed, when those tumors spread to different parts of the body. And the worst part is that cancer cells can actually trigger mutations in healthy cells. So cancer cells can cause healthy cells to become cancer cells also. And the biggest thing, or the biggest criteria for cells, is that they're supposed to stop dividing when they're too crowded. So imagine if you fall down and you cut your hand. there's going to be tissue damage and there's going to be cells that are damaged or destroyed during that injury. So when the body starts to repair itself, there comes a point where all of the damaged or destroyed cells have been replaced by healthy cells, and then the repair stops, right? It doesn't just keep growing and growing so that you get this weird, like, growth coming out of your hand. And so that's one of the, what do you call it? I don't know. I can't think of the word at the moment, but that's one of the fail safes, I guess, to prevent cancer from occurring. A normal mammalian cell stops dividing after 40 divisions. So in mammals, which we are, one cell will divide 40 times and then it will enter G0. A cancer cell is going to keep dividing even when they're densely packed, even when they're not attached to other cells. So these are cancer cells that are growing, and you can see, especially in the upper right hand corner, they're getting quite crowded, but they keep dividing. And there comes a point where probably around here that they should stop, but they just keep going and going. And that results in a tumor. Now, if the tumor is malignant, then that results in something called metastasis. That is cancer cells that spread out beyond their original tumor. When this happens, the cancer cells can invade healthy tissues anywhere else in the body because they enter the bloodstream. So if the original tumor started in the liver and metastasis occurs, then tumors can end up in the stomach, in the brain, in the lymph nodes. It can end up being in the lungs, any part of the body where cancer cells eventually settle and start forming new tumors.
A normal cell will divide only when it receives a chemical signal telling it to do so. These signals are interpreted in the nucleus, and the cells reproduce their genetic information and divide into two identical daughter cells through a process called mitosis. Cancer cells do not obey this rule and will divide even if they do not receive appropriate signals. In addition to the signals that normal cells receive telling them to divide, they are also told when to stop dividing. This prevents too many cells from being made. In fact, the cell division process is a highly ordered process. This is a critical issue in cancer because cancer cells do not obey or require normal signals for division. Normal cells will not divide when they receive stop signals from their neighbors. Cancer cells ignore this communication and continue to reproduce. This can lead to the formation of a mass of cells that piles up and may form a tumor. Also different from normal cells is the ability of cancer cells to continue dividing indefinitely.
So that means they never stop dividing. So if a tumor is operable, let's say this is the tumor. If the tumor is operable, what the surgeon will do, let me find the spot. The surgeon will measure what they call margins. And what they'll do is they'll cut out a certain portion of healthy tissue around the tumor, because if, let's say, for example, you just go and try to cut out exactly the tumor, but you can see I'm missing a little bit of the edges. What happens then is that if you leave cancer cells behind, they're just going to continue to grow and form a new tumor. So this is when, if you ever watch medical shows, sometimes they'll say, oh, I got clean margins, and that means that I've got healthy tissue around the entire tumor that I took out. Now, since I've mentioned quite often that there are a series of factors that affect whether or not someone will develop cancer, there are some, let's say, lifestyle choices that can speed that process along or put you more at risk of developing cancer. Obviously, cigarette smoking is one of them. There are chemicals or substances that are called carcinogens. These are any substance that increases the risk of cancer. So just to hopefully scare you off from ever trying a cigarette, you can see what all of the different chemicals, and this is not even all of them, by the way. So we've got arsenic in cigarettes, which is poison. We've got methane, which is what makes the sewage smell gross. Carbon monoxide. You got methanol, which is rocket fuel. There's even paint in there. Ammonia, nicotine. Nicotine is what makes cigarettes addictive, but actually nicotine is a chemical that plants produce as an insecticide, because obviously insects eat plants. And so if a plant produces a chemical that's toxic to insects, then maybe the insects won't eat it. You also have toluene, which is an industrial solvent, also bad. Hexamine, which is barbecue lighter. Stearic acid, which is candle wax. Cadmium, that's in batteries, and butane lighter fluid, and acetic acid. There are, I think, over 100 different chemicals in a cigarette. So examples of carcinogens are tobacco, also asbestos. Asbestos was used, I think, in the 1940s or 50s, maybe even earlier than that, as a flame retardant. Basically, it was kind of like an insulator against fire. So a lot of buildings, including in schools, they used to line the ceilings, I think, with asbestos. And then they found out later on, oops, it's a carcinogen. So there was a huge movement, I think in the 80s, to remove asbestos from public buildings. And radiation, of course. Most of these are mutagens. These are substances that cause mutations in DNA, and therefore may put you more at risk for cancer. Now, you might not know, but there are also some viruses that can cause cancer. In these cases, the genes inside the virus are actually oncogenes, and they can cause mutations in our proto-oncogenes or tumor suppressor genes. A very famous one is called HPV. It is human papilloma virus. Papilloma virus. Now, anytime that you see OMA, that means it's a cancer. So HPV is actually an STI. It's a sexually transmitted infection. People, like health professionals, estimate that of the entire world's sexually active population, 80% of them have had this virus. Now, the normal symptoms of HPV are it causes genital warts. A wart is basically a skin bump, and genitals are obviously like penis and vagina. So you get warts on your penis or on your vagina. But for males particularly, it's almost always asymptomatic, so you don't even know that you have it. For females, the reason why it's so dangerous is because this virus mutates cells in the cervix, and it causes cervical cancer. Now, the cervix is the opening to the uterus. So your uterus is here. This is the vagina. This is the uterus. And then you've got your fallopian tubes and your ovaries. That's kind of a small ovary. So there's your ovaries. This is where the fetus develops. This is the where pregnancy occurs. And of course, I guess we all know what a vagina is, but the cervix is right here. So that's the opening. So it mutates the cells around here, and that causes cervical cancer. And the only way that you know that you have it is by going to the gynecologist and getting what's called a pap smear. So what they'll do is they'll go in and they'll scrape a little bit of the cells off from the cervix, and they go and they test it to make sure that they're normal. So either the virus genes or oncogenes, they cause mutations, or they might activate the cell's own oncogenes by turning our proto-oncogenes into oncogenes. I think that's risks of developing cancer. And again, it depends on a lot of factors. Some families have a higher than average rate of cancer. This is what we call genetic predisposition. Predisposition means you're already at risk. So I would highly suggest that you ask your parents if they know, if their family members, if you have a history of cancer in your family, particularly certain types of cancer. For females, breast cancer has been shown to be highly genetic. So if you have a history of breast cancer in your family and your, well, actually men get breast cancer as well. I think 25% of all the breast cancer cases are men. Just because you don't have large breasts doesn't mean you don't have breast tissue. And the reason for that is because all embryos start as a default female. And during a certain point in the development of the embryo on the Y chromosome, there's a gene that turns on, and then that's when the testicles develop, the testes. So basically we all start female, yay. And that's why males have nipples, even though you don't ever breastfeed. So the more you know, yeah. Right. So yeah, I would recommend asking your family if there's history of cancer in your family. There are some genetic tests that can be done, particularly for breast cancer. Shoot, what's that called? BRCA? I think it's called the BRCA gene. If you have a history of breast cancer in your family, there is a genetic test that can be done. I think it's called the BRCA gene. And if you test positive for that, that means that you are a very high risk for developing breast cancer. Oh, sorry, it's an abbreviation, BRCA. That's what they call it, BRCA. Yeah, so there's BRCA1 and BRCA2. These are actually, I'm just Googling it now. I didn't know, but they are tumor suppressor genes. So if you have mutations in your BRCA genes, then you can keep an eye on whether or not you have a risk of developing cancer. For carcinogens, again, sorry about the text being cut off. Carcinogens, it depends on how much exposure you have had and for how long. I'll give you a personal story. I started smoking when I was 19 years old, and I honestly can't remember how long ago I quit, but it was, I want to say maybe like 12 years, 13 years, somewhere around there. So let's say, I think I smoked for around 11 years. I think until I was about around 30 or 32, something like that. And most people find it very difficult to quit once you start. It's very psychological because you're always like doing things with your hands. It's a very like hand to mouth kind of routine. And it's also like certain activities will trigger like, I need to go have a cigarette now. For me, I was very, very lucky that I just got to a point in my life where I literally just looked at a friend of mine who also smoked. And I was like, this is my last pack. I'm done after this. I don't know what it was. It was just a switch that went off in my head. And I just was like, I don't want to do this anymore. I am extremely lucky in that respect. My husband quit for, I think, about 7 years and only recently started again. It is very, very difficult. So I highly recommend don't ever, ever, ever start, because yeah, if there's like one sure way that you want to put yourself at a very high risk of cancer, it is smoking. Now, the last thing is the kinds of cancer. Cancers are grouped according to the types of tissue that they affect. And this is always for the original tumor. So carcinomas, again, remember the ending oma indicates this is a kind of cancer. Carcinomas grow in the skin and tissues lining the organs of the body. This is epithelial tissue. Epithelial tissue. So epithelial just means like a lining, whether it's lining the outside, meaning your skin, or lining your inside. So lung cancer and breast cancer are examples of carcinomas, obviously skin cancer as well. So this is an example of a skin cancer. You can see here the tumor inside the lung, and that is, it's called a mammogram. Basically, they take the breast and they squish it in between two plastic plates and basically take a high resonant, what do you call it? High resolution X-ray kind of. I don't think it's an X-ray, but it's something like that, like MRI, X-ray, some kind of imaging format. And then you see where the you can see in between the lung cancer and the breast cancer, it's kind of easy to spot tumors because they appear really dense. And so they appear more white on the X-ray. And the reason why they appear more white is because they are dense. They are dense because they are masses that should not be there. You have sarcoma, which infects bone and muscle tissue. So you can see here in the bottom of the knuckle of the ring finger. I think they're fingers. They don't look like toes. They're pretty long to be toes. And you have lymphomas. These grow in the lymphatic system. Now, the lymphatic system is kind of like a drainage system for your tissues, because as you move around throughout the day, you have fluids that build up in your tissues. If you've ever like been on your feet the whole day and then you feel like your feet are really swollen, or if you are on long flights, say like 12 or 15-hour flight, and you don't get up and walk around, sometimes your feet feel like they're like your feet and your ankles are really swollen because you have tissue that builds up, fluids that build up in your tissue. The lymphatic system is a system of vessels that brings that fluid back to the blood, but you also have lymph nodes. And lymph nodes, if you've ever been sick, which I'm sure you have, sometimes you might notice like just under your jawline, it's really, really tender. That's because you have a high concentration of lymph nodes just underneath your jaw. You have high concentration of lymph nodes in your armpits and also in your groin area, like where your legs kind of come out from the bottom of your body. Now, I had a mammogram recently, my first one, and they noticed that my lymph nodes in my left armpit were swollen. And I asked them if that could be because I got the Pfizer, the second injection in my left arm. And because vaccines are there to create an immune response, basically prepare your body for the virus before you actually get the real virus. They said, yeah, that was probably it. So there's tests they can do to see if the lymph nodes are swollen. Okay, just in time.